Welcome to those of you joining us live on YouTube. This is Genes, Destiny, and Aging with our speaker, Catherine Weir. This class is brought to you by Osher Lifelong Learning at George Mason. If you have interest in OLLI at George Mason, please visit our website, olli.gmu.edu. Okay, Kathy.
Thank you. Welcome back to number 4 of Genes, Destiny, and Aging. I'm just going to quickly review by going over the course outline. We've been talking about research findings about genetic and environmental influences on different human behaviors or traits. In the first week, we talked about early studies, actually from 600 BC, when Samtic was trying to figure out whether there was a genetic predisposition for humans to use language, until nowadays. We're going to have quite a few references from 2020. One thing to remember from that is 98 or 99% of the genetic material is the same for all humans. That's what makes us human. And so any differences are all going to be focused on that little tiny percentage of our genetic material. In weeks 2 and 3, we talked about what the behavior geneticists do. What their main enterprise is to estimate heritability, the proportion of the variance that is due to genetics for any particular behavior or trait. And in doing their studies, mostly their twin studies and adoption studies, but they also use other kinds of family studies, they also estimate the shared environment. And by that, they mean the rearing environment of an individual being raised in a particular family. Today, we're going to talk about nature and nurture working together, sort of combination effects, and a new approach that started about 2007 called the genome-wide association studies, the GWA. One of the things to keep in mind is if you're trying to remember what is GWA, I remember it as since we're so near George Washington University, GWU, but the A comes ahead of it. So, but it really stands for Genome-Wide Association Studies. The dates of this starting 2007, the behavior geneticist enterprise started at about 1970. In fact, there's a journal called Behavior Genetics, and that was volume 1. Well, let's get started by evaluating the behavior genetics approach. Just as a reminder, and this is something we ended with last week, a particular example of behavior genetics for the propensity to marry. It was based on research by Jersky et al., and they had 6,300 pairs of twins in the Vietnam registry. And for any behavior geneticist, you start with a model or conceptual equations. The main ones are for twins are that monozygotic, that's identical twins, raised together, that's the T, they should share 100% of their genetics, and they should share the shared environment if they're raised in the same household. The way they come up with that conceptual equation is by knowledge of genetics and how genetics are passed on from well, in the case of monozygotic twins, the 2 eggs separate after conception. So that's why they share all their genetics. Dizygotic twins, the fraternals raised together, they're like ordinary siblings. And we showed that it means that the siblings share half of their genes, 0.5 of the genetic piece, and the shared environment. the way they estimate heritability, usually there are a couple of ways. The way we talked about the most is an easy way that we can just do without having a whole lot of computer programs to do statistics behind us. And you simply correlate twin partners. In this case, since we're looking at propensity to marry as our variable that we're the psychological variable, you take the ones where both twin partners are married or both twin partners are unmarried. And the other side is the discordant side, where one of the twins got married and the other one remained unmarried. And you can work from a tetrachoric correlation. It's not the same as the R we talked about in the second class. In this case, you have different statistics behind it, but the tetrachoric correlation can be worked out. And you take the identical twins raised together, the correlation between them on whether they got married or stayed unmarried versus whether they had discordant desires about marriage. And you subtract the fraternal twins raised together. And when you do the math with the conceptual ideas based on genetics, you get half of the heritability. And without too much fiddling around, it's easy to see twice the difference between the two correlations will give you G. The other way that behavior geneticists do this, and this is much more common nowadays, is they have a model of how they would predict genetics has an effect, shared environment, non-shared, on the propensity to marriage. And they evaluate which model works best. I mean, one model might be that there is, it's all genetics and nothing coming from environment. And they try that out and they see if their results come out anywhere near looking like that model. They pit several models against each other. Well, in Jersky's experiment, they found for the genetic, the heritability, the genetic proportion of the variance was about 41%, and shared environment was about 15%, leaving the environmental things that happen not in the family to everybody, but uniquely to each individual as non-shared. And in fact, this probably should be called residual or all the rest, because any error in measurement would be added into this. And that one comes out as 0.44. So you can see that the shared family environment is much less important for a propensity to marriage than either the genetic component or the unique things that happen to individuals, even though they're raised in the same environment. Well, when you're looking at an evaluation, you want to look for strengths and weaknesses. And the strength of the behavior genetics is that we can measure the percentage of genetic and shared environmental influences for any trait or quality that we can measure. In fact, reliably, I've put here, you can actually work out how reliable your measure is. In the case of getting married or not getting married, as long as you have accurate records, that's a matter of legal standing, and that shouldn't have any error of measurement. But most of the things that we look at, we don't measure precisely. When studies were done with twins and adoption, IQ came out for the heritability around 0.52, about half of the variance of IQ came from genetics. Openness, this was one of those personality scales in a standard personality test. At one end of a continuum was people who wanted to keep everything just the same. And at the other end of the continuum, it was people who much preferred everything being new all the time. And they too had a heritability in the 0.5s, a little bit more than a half came from genetic sources and influences. Whereas with political attitudes, it was different. The political attitudes that are normally studied are where people fall on a continuum between conservatism and liberalism in the political science definitions. And if you look at 17-year-olds, they have only 4% that comes from genetic sources. But if you look at older twins, this is a case I looked at 67-year-olds, they also had a genetic contribution of about 54%. So we are able to put a quantity for how much of a particular behavior comes from genetic and in different environmental sources. Well, let's look at the weaknesses. That was a benefit. One weakness is twin studies. One of the first problems, if you're going to do a twin study, is to assign twins. Are they monozygotic or dizygotic? And you could be wrong. Sometimes if you just look at siblings, not twins in the same family, they look very much alike. And that would happen also with twins that actually were from separate eggs that were fertilized by separate sperm. That's what makes a dizygotic twin. So actually how you go about assigning them is quite tricky, and it can be very expensive if you're going to look at the DNA. Another problem with twin studies is the environment for twins as children might really be quite different from that for singletons. And twin births are relatively rare. It's kind of hard to find twins compared to singleton. Even with a lot of twins being born now more than before 1980s with fertility treatments, we're still not talking about more than 3%. So singletons are 97% of the births. Another problem that the DNA of monozygotic twins is not completely identical. It turns out that some of the base pairs on the DNA mutate, and this small number that mutate, that starts within days of conception. And what happens is, we got to remember though, that there's 3.2 billion base pairs in human DNA. So the DNA of monozygotic twins, the same egg that for some reason splits into two different individuals, is much more similar probably than the two separate eggs that are fertilized by separate sperm. Even so, there's data now showing that it's not 100% genetics. That's getting at one of the assumptions. There's going to be errors of measurement in all experiments. Like intelligence tests or different kinds of cognitive mental testing tend to have an error of 5 to 10%. Personality tests, the best ones, have an error as big as 35%. So that the second time a person takes the test, they might not come out with the same value on a particular scale. The other problem with any kind of measurement is you get a particular sample on a particular day. It's the problem of all statistics. The wording of questions or instructions might turn out to be critical. And we make lots of assumptions when we work out heritability, and it could be that we just have a bad question or a bad questionnaire. A third problem is based on adoption designs. One of the points that certainly in developed nations, adoption is selective. Families that are allowed to adopt are nurturing, and most of them are middle socioeconomic class. And it's not done at random. when people have done studies to look at this, they've said what's different between a middle class environment and, say, a lower class environment. And I'll give you just two problems. In a working class environment, in the first few years, some psycholinguists have counted 20 million words are heard in their experience as young children. But in a middle-class household, it's 30 million words. So exposure to language is going to be different. Perhaps a particularly important one is some people studied how many encouragements are given to children dependent against how many reprimands are given to children. And it turns out that working class parents give twice as, 1/2 as many encouragements per reprimand as people in middle socioeconomic class. That's not necessarily for twins or anything, that's just in general. Another problem about adoption designs is you cannot always control the age of adoption. If the adoption happens at 5 years or at 10 years, that's going to be very different than the baby who's adopted at 1 month or less in age. And in a lot of adoption designs, there's an effort to pick up like a biological parent with an adopted away child, or 2 biological siblings, one that went to one family, another that went to another family. And this can end up with very small groups of adoptees, because actually tracing the biological parent or the biological siblings is very complex. Ploman reports an IQ study with about 5,000 identical twins and about 5,000 fraternal twins, but all they could find was 800 pairs that were the biological parent with the adopted away child. So any estimate you pick up from adoption designs is probably going to be less accurate than a heritability or environmental influence that's based on much, much bigger samples of participants. Well, what about assortative mating? The behavior genetics model assumes that mating is random, resulting in offspring. And David Buss, back in 1983, I think it is, was interested in what you hear in sort of People magazines, do opposites attract? And he looked at correlation between parents. And what he found is there are a lot of positive correlations between different characteristics of parents. Their ages correlate around 0.8, their educational level around 0.6, their ethnicity 0.6, their attitudes 0.5, mental abilities 0.4. And as we're going down, social status 0.3, other aspects, physical height and eye color, about the same. Personality variables actually correlate the least in the set that Buss looked at. So it isn't like people, a male and a female are randomly assigned to be mates. And David Buss in 1989 did a pivotal study where he had data from 39 countries with a survey that was like this. Based on the question, for a long-term partner, rate how important these characteristics are for you. And what in his case, he allowed people to rate. One was it was essential that a long-term partner did not have a characteristic. To 10, this was a much preferred characteristic. And I'm just listing 6 of the ones he used. He used much more like 15. If that partner has an exciting personality, or if that partner has good earning capacity, or good health, good housekeeper, kind and understanding, or physically attractive. And he asked people to rate the importance of these in a long-term partner. I thought it would be kind of good for us if we did a little poll, but we don't have time to repeat what Bus did. What I've asked you to do is, out of these 6, when you get to the poll, to choose the one that's the least important to you, not the one you prefer, but the one that you would think is least important in a term of a long-term partner. It doesn't, people sometimes ask, does it have to be my real partner? Or the answer is, if you like, it can be your real partner. If not, it can be some ideal partner that you might have a long-term relationship with. There's one other question in that poll, which is about heritability, and I'll let you read it. Okay, can you pull it up, Meg, please? Or shall I try? There we go. I'll give you about 20 more seconds. So let's look at, I'll share the results with you. Heritability, or G, tells us whether a particular individual has inherited a trait. The answer should be false. So it makes me realize that maybe I haven't taught very well, but I'm pretty sure by the end of today, you'll get a better feel for it. What happens is that it tells us about the variance, the variability among individuals in a group. How much of that is related to genetics? So it doesn't relate to a particular individual. On the second one, which of the results, which of these 6 items do you think is the least important? And most people have answered good housekeeper. Although if you look at marriage research, a lot of arguments about who's tidy and who's a slob. But exciting personality looks like the second most important. And in some studies I've done with lab classes at Colorado College, we found that older people, and I'm assuming most of the people in this class are older, exciting personality was much less important. And when it was compared to the young people, they much preferred exciting personality relative to old people. And so I'm going to move away from that poll and show you what was the result of David Buss. And he was asking, are preferred characteristics of long-term mates universal? I'm going to explain that in a minute. There's a recent study. David Buss is one of 108 authors in it. It was international. People reported similar preferences and characteristics for long-term mates across 45 countries. This was based on 14,500 people in the age range 18 to 91 years. What was organized was a big group of researchers in different countries, and they had to get at least half of their participants from the local community and half from the university. The average age of respondents was 30. And what were the main results? They're actually illustrated here, but as in 1989, when you compare men to women, and that's what this graph is trying to show you, if the answers are on this side, females thought it was more important than males. If it's on this side of the line, males think it's more important than females. And the result in both 89 and 2020 is men rate physical attractiveness as more important than women. And that's why we see physical attractiveness over here on the right side. Whereas women rate financial prospects higher than men. And that is the yellow here. And they got that same result with the 45 countries. The difference over here with age choice, it turns out that when asked about preferences for age differences, older men prefer younger women and younger women prefer older men. It turns out both ways, and that's what's being illustrated here. In any case, we can't say that marriage or long-term mates is a random process. It's very orderly, and it appears in many countries all over the world. I did look at the list of countries. All the continents are represented, although Africa is only represented by Algeria. But many, many other countries in the East, Middle East, Europe, South America, Central America, North America. Well, so that's another assumption that's not met. That was part of when we generated the behavior genetics equations about how similar identical and fraternal twins would be to each other. One of the things we've seen frequently is that genetics and environmental influences change with age. And there's no provision in that in the behavior genetics ideas. We looked at this evidence at different times. If you look at IQ, what happens is the heritability starts at around 0.22, and as we get older, it goes up. In this particular sample, by 75, it was down to 0.58. But in many other samples that have been done later, it stays up around the 0.8 level. So there's certainly an age trend. At the same time, the importance and influence of the shared family rearing environment turns out to follow the opposite trend. For young kids, it's pretty important, and it gets less. And by the time kids get to be 18, and in our society anywhere, leave high school, it becomes very small indeed. And what about political attitudes, conservatism to liberal? Up to 20 years, there's virtually no heritability in the political attitudes on the continuum between being conservative and being liberal. Whereas shared environment, though, is quite large. I haven't given actual numbers, but this is the basic trend. Over 20 years old, when the participants, suddenly you see there is a genetic element, and it's around 0.5. The shared family environment, the rearing environment, remains, there's some, it's around 0.15 or so for females, but about 0.02 for males, for your rearing environment, how it had an effect on your political attitudes. For personality scores, they too change with age. And this was a study reported from 4 countries, Germany, Croatia, Finland, and the UK, with 7,000 twin pairs. And what they gave personality tests, this one had not 5 factors. Emotionality was, one end was to being really laid back, and the other end on the emotionality scale was being hypersensitive and anxious about things. They have extroversion, introversion, agreeableness, non-agreeableness, being conscientious and being considering things just to be trivial. And openness is whether people like something new better or do they prefer the old. And honesty, humility was the additional, in addition to the 5 factors that we've already talked about. And what you can see is there are a number of age trends here. What they've represented in, let's look at red first, the heritability. You can see that honesty, heritability is important for 14-year-olds, but by the time the twins are into their 70s or 80s, heritability isn't nearly as important. Whereas shared environment becomes more important. That's the blue. If we look, for instance, at, well, what would be another one? Agreeableness, the genetic aspect of agreeableness goes down, whereas agreeableness, the influence of shared environment, sorry, this is the non-shared environment, and please eradicate my thinking about shared. This is the non-shared, where things that happen to individuals stays about the same throughout a person's life with being agreeable or not agreeable. And you can see a great variety of age changes, none of which are accounted for in the basic models for behavior genetics. A 6th challenge of the behavior genetics approach is when genetics and environment work together in combination. For those of you with a statistics background, this is called the genetics by environment interaction. It's a combination effect. Genetic predispositions might influence environmental events. The person who is extroverted might influence environmental events very different than the genetics for the person who, a person who is more inclined to introversion genetically. And it works the other way too. Environmental influences, the way the genetics influenced ability. So let's look at how this can happen. Scar and McCartney back in the early 80s talked about 3 ways that we can have a combination effect of genetics and environment. One they talked about was passive. When a person is exposed more to a certain environment by the parents, remember the parent and the child share 50% of their genes. What's an example here? Well, let's say a child is raised with parents who enjoy reading and who talk a lot about the books that they read. They expose the child to the world of books, take them to libraries, have lots of books in their own home. That would be considered a passive way that genes and environment interact. An evocative way would occur when someone else, not the parent, but someone else in the environment works to enhance the genetically influenced ability. Perhaps a coach for a soccer team is allowed to award a scholarship for soccer camp. Who does that coach choose? The kid that has the most coordinated abilities, which have been found to be genetically influenced. It's as though the coordination for that team evoked the coach to give the scholarship. Then, of course, if the kid goes to soccer camp, they build their abilities more by just practice. A third way is the active genetic and environmental combination effect, where a person chooses an environment because of a genetically influenced ability. like a musically able person joins a music group, and then that means they practice it more again. So you can see what we mean by both genetics and environment working together. Why is this a challenge to the behavior genetics models? Well, I have up here the variance of the IQ was done on 12-year-olds, and we could see that about 52% came from genetics, and about 20% each was in shared environment and in the non-shared environment. And there was an error of about 0.08. This was for IQ. But if you had a case where, let's say, low IQ parents expose a child to an environment with only low mentally challenging tasks, there's a passive genetic effect. I wanted to illustrate it could be on both ends. Like we gave the illustration where both parents do a lot of reading. Now, this is a case where probably some of those cognitively unchallenging tasks like watching TV is the main entertainment in that family. The question is this in the child's shared environment, or is it because of the genetic influence? Because the parents had low IQ, and their kind of time in the environment is spent doing low challenging tasks. These are what we call non-additive effects, because the whole idea of the behavior genetics model is you can take the variance of anything you can measure like IQ, and you can find separate bits due to genetics, shared environment, non-shared environment, and error. But it's not additive if it should really fall in two of those cells. Do we count this bit, this passive genetic environmental effect? Do we count it in the genetic side, or do we count it in the shared environment side? If we count it for both, we have more than 100% of the variance allocated. And so the combination effects challenge to the extent that there are non-additive effects. And the recent use of behavior genetics separately work out the non-additive as opposed to the additive effects for whatever it is they're measuring, IQ or personality or other things. I want to illustrate this again. This was it's known as a candidate gene study by Caspi and a number of other authors in 2003. They were interested in understanding how much stress and depression are related to the serotonin transporter gene. So they noted that depression is a leading cause of disability. And they thought, maybe what we'll find is a difference between depressed people and people who are not depressed with the serotonin transporter gene, which is called 5-HTT. It's on the 17th chromosome. It's this particular location on that chromosome. And it's what Prozac does. When you take Prozac, it changes the rate at which serotonin is taken back into the system. And serotonin is known to control things like mood, memory, reward, learning. And it turns out that people have either a long allele, that's an allele is a particular little stretch on the DNA, in this case, on the 17th chromosome. It can either be long, which means there's more reuptake of this serotonin that makes for a better mood, or a short allele, which has lower efficiency. If you actually look at the base pairs, the length of the allele is determined by how many repeating codes there are the end. So one is long and one is short. let's see here. They formed 3 groups of subjects because it's a person either has 2 short alleles in this particular location on their DNA, a short and a long, one from each parent, or 2 long alleles. And you can see about half the group had 1 short and 1 long. They got about 847 participants. These aren't twins. They're in New Zealand. They were Caucasian, but they weren't Maori. They averaged 26 years old. And they did a standardized depression interview by professionals, which included a questionnaire about a self-report questionnaire, and an observer, somebody known to the person who observed the behaviors, and they did DNA sequencing on all of these people. And they, in that particular group, 17% had actually had a past year major depressive episode. That was 144 people. Well, they were interested in studying stress with depression. So they have to measure stress. And can stress cause depression? Well, there is a stress life events questionnaire. It asks questions like, have you had a change in job in the last year? Financial issues, have you had a windfall, or have you lost a lot of money in the last year? What about your housing? Have you had to move house? Have you been evicted? What about your health? Did you have a diagnosis that made a big difference in your life? Or did you have an unremarkable health during the last year? What about a relationship stress where perhaps there was a breakup of a long-term partner, or even a marriage? They all count as stresses on the life events questionnaire. And this was the findings that CASPIF had. They have down on the horizontal axis the number of stressful life events. A fair portion had none. Some had as many as 4 different life stresses. Maybe lost their job, lost their relationship, maybe had got into financial trouble, and well, let's make it 3. I can't think of 4 offhand. And in their self-reported depression symptoms, that's what's on the vertical in this section A here. And what they found was that the people who had 2 short alleles actually had more symptoms that they reported for themselves of depression as we increase the number of stressful life events. The people with 2 long alleles increased, but not nearly as much as those with short alleles. And the people who had 1 from each parent, a short and a long, fell in between. In B, they measured the depression episode by the criteria used by professionals, and they got essentially the same thing. In D, what we see is the informant, the person, the observer, noting the behaviors. And again, you see that the genetics made a difference how stressful life events affected the individual. This last one is the probability of suicide, thinking about suicide or attempting it. And you can see as the people with 2 short alleles, it rises particularly fast as we move from 3 to 4 stressful life events. That's a combination effect of the genetics with the number of environmental stresses. And again, it's going to cause a problem for the behavior genetics approach. The 7th challenge is that genetics and environment are only gross estimates. There's no specific mechanism of how the genetics has its influence on the behavior, like depression or on the political attitudes. 50% genetic or heritability doesn't explain how particular genes influence intelligence, personality, or social attitudes. And 10% shared environment doesn't explain which household or family factors accounted for a particular person's GPA or their depression or whether they decided to be divorced. Also, this is what I was trying to get you to think about in my poll. It turns out that estimates of heritability G and environmental shares of is about the variance of a group, the variability of a group. It isn't about individuals. And we can't say anything special about individuals. Well, in between, let's say, 1980 and 2005, there were a number of gene hunting or candidate gene studies, like the Caspi study I just showed you, that related depression with the number of environmental stresses, the genetics of depression. And it turns out it's often called a candidate gene fiasco, because if you think of how many genes humans have, and if you're going to test just one or two, you'd have to be incredibly lucky to actually find that little stretch of DNA that was important. And most of the studies didn't work. Caspi is almost always suggested as the one that showed what ideally happened, but it was probably because they were just lucky. And I want to remind you, humans have 3.2 billion base pairs in their DNA. To pick out one or two is incredibly, maybe based on some facts, but it's going to have a lot of probability of failure. We said that if you look at the DNA, it's like a ladder with rungs, and that's where the base pairs are, the rungs. And 99% of the rungs in the DNA ladder, that's the nucleotides, are the same for all of us. If 1% of the rungs differ, that still gives us 60 million nucleotides. And I'm talking here about the coded part, the codes that the DNA has that eventually contribute to making proteins like neurotransmitters or skin. And when cells divide or replicate, a few mutations occur, even though our system, and it's presumably because of the DNA itself, have many, many quality control mechanisms. I went to the talks on cancer, the Emperor of Diseases in the fall semester, and I loved the analogy that Suresh Mehta made about it's like the barcodes that if you go to a self-checkout, they need to be exact to decode how much the product is that you're putting into your own bag. And so it is. There are a lot of quality control mechanisms. In fact, I think Mehta gave the example that at least 80% of the time when a cell is dividing is spent double checking the quality control. But even so, we're talking about 60 million nucleotides. And human individual differences happen because of the number of ways that human DNA sequences can vary. It's really a mind-boggling big issue. So just to review some of these evaluative points I've made, the benefit of behavior genetics is that we can get quantities for the genetics and the environmental influences, so we can focus other kinds of research better. But the challenges, mostly they're about the assumptions of the models, are problems, practical problems of identifying twins and knowing exactly what their DNA tells us. That adoption is selective, not random, which was part of the assumption. Mating is assortative. We don't randomly assign partners. And one of the things that comes up over and over is we see changes in the heritability and the components of environmental influences for different ages of participants in the studies. There are a lot of genetic and environmental combination effects, the non-additive ones, and we're only getting gross estimates of the variance when we actually follow through with the twin and adoption and family studies. The last piece here is that gene hunting, though interesting, has rarely been successful. And it's quite expensive to do a lot of these studies. So you need to try to get better bang for your buck. Now, I'd like to talk a little about the genome-wide association enterprise. Why have a new kind of method, a whole new enterprise? Well, it started with dissatisfaction with the finding that heritability and environmental components for any particular behavior were just gross estimates. And it only applied to the variance of the group, not to individuals. Also, it wasn't connected in any direct way to the DNA. So those kind of bits of dissatisfaction led people to make a new kind of method. Also, it was fed by advances in biology. For instance, we understood more about the chromosomes having DNA, and that how the DNA gets out of the nucleus and via messenger RNA moves into the outer part of the cell, and how that together encodes particular amino acids, which in combination make proteins. And technological advances also have made it affordable to study DNA. But if you're going to be in the genome-wide association method, you're going to have to be able to think big. You need big groups of researchers working together, big studies of many individuals from many countries. You need big financial backers. And there's always been a lot of special interests in medical applications. I might add that a lot of these studies are now not taking place in the USA because we don't have a nationalized medical system where we can get anonymous data that is kept in big databases. Almost all the data that we're going to be getting here, you're going to see, is coming from other countries where there's some nationalized medical facilities like Canada, Europe, China, and most of the Asian countries. Well, the results of many genome-wide association studies. I'd like to give you what's going to be the result when we get started so you can keep it in mind. The differences in the nucleotide bases in the DNA of individuals show that each chain in the base pair has a very small effect on the behavior. So if your DNA has one base pair that looks different than the person next to you, it's going to have possibly a very small effect on your IQ or your personality trait or your political attitudes or whatever is being measured. And one of the things that's going to come up is that future research will need to fill in many gaps in our knowledge of how genetics and environment influence behaviors. So far, the nucleotide base pairs that distinguish people individually from others account for much less of the variance of a behavior than the heritability estimates of G. So let's get started on some of these. Well, mostly we study where there's just one base that's different than other people's. It's the most frequent type of difference among humans, and it's called a single nucleotide polymorphism. And if you take the first letters, SNP, people pronounce it as a SNP. It means it's simply that in the sequence of DNA in one person, one particular nucleotide base is different from the DNA of others, or at least of some others in a population. So for instance, at a given location in the DNA, some people, if you'll remember, there are 4 possible nucleotides: adenine, trypsine, and in some people it's an A, in other people it's a T. And in fact, T is the most likely base in the sequence in that particular location in the DNA. So if the sequence around is, let's say, a C cytosine, and this is for adenosine and this is guanine, codes for the amino acid valine. We talked about that. It was before. It's a particular amino acid that when mixed with a lot of others, functions to change what goes on inside a cell. But if one change in that particular location in the DNA, one base is changed, so it's a, say, GAG, then a different amino acid would be coded for, leucine. And this can change many of the ways that proteins are formed, because proteins are made up of many amino acids. The protein itself might regulate different processes, like your neurotransmitters or the formation of immune responses or the way insulin works. It's known that humans have probably 4 million SNPs. That's where one nucleotide base is different, but many occur for only a very few people. But you have to remember that in humans, we have 6.4 billion nucleotides in the DNA in each cell, 3.2 from one parent and 3.2 billion from the other. So the known single nucleotide differences account for less than 0.1% of the differences among individuals. It's a lot of hard work to be done to understand it. I want to give an example that Plomin gives in his book called Blueprint, a SNP example that influences body weight. There are many different single base differences that have small effects. If you remember, we talked about that human chromosomes can be put in fluorescent microscope after they've been marked with a fluorescence chemical. And it turns out they can be put into a lineup called a karyotype with the 23 pairs chromosomes. A polymorphism, that was the word where P comes from. It means a base that differs among people. It can be a different nucleotide, like C instead of A, or it can actually be the different length of a repeat of the DNA, like we saw in Caspi's case, where people with 2 short alleles tended to be much more anxious and more likely to show effects of stress than people with 2 long alleles. The single nucleotide difference is on chromosome 16. It's down here. Remember, they're ordered by their size. It's usually T at the locus of the 50 3,767,042nd location out of the 90 million bases on that chromosome 16. Some years in the past, this mutated from T to an A in an ancestor. And now if you look at the population, about 40% of people do have A in that location on chromosome 16. 60% still have T in our human population. The name given for that one nucleotide base difference is FTO, the name that the geneticist gave. As Plomin says, thank goodness it's going to be about body weight. They didn't call it fatso because that's what it encodes for, or one of the things. And what it actually codes for the protein that accelerates chemical reactions. In a large random sample of people, each A is actually associated with 3 pounds more weight for that person. And you get the regular kinds of genotypes, which you probably learned about in high school. You either have 2 A's, an A and a T, one from each parent, or 2 T's. T was the one that it started in our human existence. A was a mutation at one time and has been slowly passed on. And we see about 50% of people have AT, 35% have the double T, and just 15% have the genotype with 2 A's. And if you weigh them, these are big groups of people, the people with double T are the average weight for that person, male or female. It would have been done separately. If the person who has the genotype with 1 A and 1 T, they're on average, that group is 3 pounds more in weight. And a person who happens to have A from both parents will be 6 pounds greater. So that's how, just an example of how this might work. Each single nucleotide base difference, that's a SNP, has many effects, and this is known as pleiotropy. One change, like an A replacing a T at a certain place in chromosome 16, can alter many different behaviors that influence body weight. We don't know whether valine or leucine, that's the amino acids that the code actually produces, can change the way that fat is stored by the person who has the A rather than the T. Or we don't know if it changes the sensation of hunger when full, which is a part of the psychological study, mostly in the hypothalamus. And the pleasure derived from eating sugar. There are many ways you can influence body weight, but we can at least tie it to the change of one single nucleotide base. Also, that a single nucleotide base has many effects, but we can see the opposite. Each trait difference, here different people's body weight, can be influenced by many different single base differences. And that's called polygyny. The nucleotide base at the FTO location on chromosome 16 is either an A or a T. And when research was done with a big group of people, it accounted for 0.7% of the variance difference between those people who were overweight and those who were of an ideal weight individuals. In other words, a tiny bit. And in a big study, they had with 97 different single nucleotide base differences, altogether, they came up with only 1.2% of the, I'm switching from body weight to body mass index, but that's connected with body weight. Whereas studies of the body mass index for 12 to 14-year-olds show that its heritability is about 54%. So that one single place on chromosome 16, being an A or T, is a tiny, tiny bit of that that 54% for the genetics. In a bigger study with people of different ages, the heritability for body mass index comes out more between around 0.74. There's a huge gap between specifying a particular locus on the DNA and the behavior genetic results. So there's a criticism of the genome-wide association studies. They only help us with just tiny, tiny bits. What are the technical advances that enabled genome-wide associations? Well, the one technical advantage was these chips were developed that test for single nucleotide differences. And you can buy them. Sometimes, here's a picture of one of those chips. You put it into a DNA sample and it can tell you what base is at 10,000 locations or up to 250,000 location for one SNP chip. And the price has gone down. At one time, to get a whole genome sequenced, it would cost about $1,600. But now with a SNP chip, you can focus on regions more likely to be important and in fact end up with only $80. I found that if I went to Google, I could simply say SNP chips and I got my options of several different places. If I wanted to get fewer than 7, if I had only 700 individuals and they had an assay in stock in this one, it would cost me $258. And I could test for, it could have been the 10,000. And it's now gotten so it's like 60 cents per SNP per sample. We're not talking huge money now. And the techie people are building more and more possible single differences. Well, researchers have to work together. And in a paper by Horwitz in 2019, they formed big working groups. 2017, the Genome-Wide Association Group pooled cases for 7 disorders. Remember, most of the money comes for health problems, dementia, schizophrenia, bipolar. People put their results of studies, in this case 2,400, into the Genome-Wide Association Study Catalog. That was 10 years after the whole approach started. The SNP chips that looked, in this case, for 28,000 different base differences. And with schizophrenia, they found 74 of the 28,500 base differences differentiated the schizophrenics in their sample of the many different studies from people who were not schizophrenic. But that only accounted for 0.3% of the variance. And schizophrenia has a very high genetic heritability. It depends on the study you're looking at, but it's around 0.8 or so. And one of the things we see here is Alzheimer's dementia, for instance. It shares links with body mass index, cholesterol, lifespan, caffeine addiction, shares links with blood pressure and metabolites. And when the researchers in that paper argued that we need to confirm these results to be sure they're accurate, but we also need to take account of where the links are to one disorder to another. This was the picture they showed of some of the links in different places. I thought you couldn't see it. I made just a small bit here. Let's just focus on the caffeine, coffee consumption. Yellow is for the psychological variable. Down here, educational attainment. Red is the particular location in the gene. And in this case, they actually tell you what the base was, adenine in that case. And for coffee consumption, diastolic blood pressure is connected to it. Also, blood metabolite levels. So you can see, oops, did I go backwards? Oh, no, I didn't. I brought in this one. Look at Alzheimer's disease. How many direct links to different genes are there? I count 7. This gene, 2, 3, 4, 5, 6, 7. And the greens are other bits of information that might be important. Well, if you take this one, we know with Alzheimer's, amyloid, plaques, tangles are important. So what's the payoff after these years of study? Well, thinking about so many single nucleotide differences have such small effects was a big jump from where we started 25 years ago. We now know for certain that heritability is caused by thousands of associations, each. Even with such small effects, it is possible to consider where a person is genetically compared to others along a continuum of a psychological trait or behavior, like along a depression scale or a scale based on the person's school achievement or their attitudes on the conservative liberal continuum. And Plomin uses himself as an example. He was compared in this case to a big twin study, including 16,000 twin pairs. They were tested many times between the ages of 1 and 21. It's not a very good control group because Plomin is now old enough to be retired, but his educational attainment would have come out genetically in the 94th percentile. So he would probably score better on his GPA than most of the others. He was a bit worried about, he came up in the 85th percentile for schizophrenia, even though he feels like he has none of those symptoms. These other are more a lower value, and each individual can be characterized using a genome-wide association. Well, let's just finish off here with going back to the study that we talked about last time. We need to identify particular biological systems that were important to the behavior genetics finding, the heritability and the environment. We test attitudes using the list of issues like property taxes, gay rights, X-rated movies, death penalty. Each person says, I agree or I disagree with these property taxes, or I have no opinion. And the final score puts the person on the conservative liberal continuum. And what we found last week is 17-year-olds had virtually no genetic heritability. And you can see here's the monozygotic and the dizygotic twins are on top of each other. But by the time they were 67, we saw a much bigger heritability. It came to 0.54. This was based on about 7,000 twins, same-sex twins. In the Hatemi study, which was actually done in Australia, so they changed the words for the issues to be right for Australia, they had 13,200 people from just under 3,000 families who did the conservative liberal test and who gave DNA. And what they found is it's an exploratory method to identify different regions of DNA that correlate with the score on the political attitude test. They suggest this reduces possibilities. And here's a picture of what they found. These are locations on chromosomes. If you'll remember, it goes from chromosome 1 to 22. 23 is the sex chromosome. These twins were same sex, so they didn't look anything on that. And the places where they get a big difference between the conservative and the liberal scores turn out to be, well, the biggest one was on chromosome 4 at a particular location. And they said, well, what could that be? And they looked at other things that have been connected to human social behaviors. It turns out that that particular location on chromosome 4 is linked to a neurotransmitter that promotes neuronal and vascular growth. It involves cognition, memory, social learning, fear conditioning, social behavior. The next biggest is over here on chromosome 9. They couldn't find any way that it related to behaviors of conservative liberal political attitudes. Chromosome 2 was the third, and they saw that as being related to other neurotransmitters that included serotonin and dopamine. So, summary of the course. There are many limitations of the behavior genetics models on which estimates of heritability and different kinds of environment are made. But gross estimates is a problem because it doesn't tie it to DNA. But the new genome-wide association methods you have to have large samples of people, large samples of DNA, nucleotide base differences, and they find that they show very many tiny, tiny changes that work together to account partially for the traits and the behaviors we measure. But researchers say that many unknowns are left to fill the gap between the heritability estimate G and the specific codes in the DNA that influence behavior. Bowman calls it the dark matter of the nature-nurture research. So anybody here who'd like to have a second career, I'd suggest that there's an awful lot of options to work on that dark matter, given that we have so many bases and the geneticists only know a little about them. So why is my course title demolished by research? Well, genes, that's a problem. This is a picture of genes. It has 4 different bases, blue for one, red for another, pink for another, and green for the last. You can see it's just a real hodgepodge, the 6.4 billion bases. Nowadays, geneticists identify a gene as a nucleotide base pair on the DNA, often only those that make proteins, that make codes for proteins. And they have been having contests as to how many genes humans have. In 1960, people thought it was going to be 6,700,000. These are the number of genes in thousands. 1990, the suggestion, 100,000. But by 2001 or so, when the first draft of the human genome was released, the numbers were closer to 30 to 40,000. But when the human analysis of the complete genome was completed, we came down to around 20,000. And remember that these are the latest count found, 21,306. All of these are the protein coding genes. And people are still arguing with Salzberg, who I think works over in Johns Hopkins, that they didn't like the way he counted. So geneticists are arguing how you can count such big numbers of things. He used a computer method. Most people use some sort of a method. Why is destiny gone from my title? Well, destiny, at least the connotation, is something inevitable, but really all we can get from behavior genetics and from the GWA, the genome-wide associations, are very limited predictions. We saw just tiny little predictions, but for a given person. Behavior genetics could give you big gross numbers, but it only tells you about the variance of an individual. Aging is left. And I think there are a lot of developmental issues, and the book called Innate that I recommended by Kevin Mitchell suggests that this kind of development may be part of that dark matter. So maybe what I should have called the course is something more like single base pairs that make proteins and aging. That's the end of my presentation here. Are there lots of questions? I will end this stop share here.
Steve Greenhouse has a question.
Well, I can see in the chat, are
Yeah.
the 3.2 billion base pairs in DNA alleles? The point is we know about a few of them, and the answer is nobody knows. They've just assumed. But the ones that are called alleles are the ones that differ when we compare different individuals. Remembering at 99%, or was it more than 99% are the same? So allele is a word that came from Gregor Mendel in the middle of the 19th century. We don't have a precise equivalence. Why does genetic heritability rise so dramatically with age? If there is a genetic influence, wouldn't you expect it to be constant over time? And what happens is that, in fact, that was the assumption of the behavior geneticists who started in 1970, that genetic influence would be expected to be constant over time, but it is not. So I do recommend, I'll send you the reading list again. I think I sent it before the course. These are books written for lay people that Kevin Mitchell has written, suggesting that that change over age may be a part of all that DNA that we used to call junk DNA, but we thought was important. If you find out, Ed Rader, you let me know.
Okay, Steve Greenhouse had his hand up for a question.
Yep.
Yeah, very interesting course, and it's a lot to swallow here.
Yes.
But I always thought that it was genes that determined either physical or behavioral traits. In other words, the traits were correlated to the genes, but I think you're saying they're correlated to the base pairs.
Yes.
And genes are maybe an artifact of a bunch of base pairs that don't, you know, you have to look at the base pairs or stretches of base pairs on a chromosome to determine the trait, and that the genes don't really have much meaning anymore, or they're an artifact. Am I confused or what?
Yeah, the suggestion I've heard is that Mendel was lucky to make something that worked like genes so that we had the dominant recessive kind of arithmetic, if you like. And yes, nowadays we're down to identifying specific places of DNA. And we then get into this business of each specific place only affects many things, not just one. And we don't have any of these nice little genes that Gregor Mendel talked about in that sense. Yeah.
But if a base pair is mutated, that doesn't mean the genes necessarily mutate, or it takes many pairs to mutate before the gene is mutated, or does that not even matter?
If it's a protein coding gene, one of the 21,000 that the geneticists are arguing about right now, they actually put bets in as to how many there were, and they raised a lot of money that way. But what happens is that that will change what is encoded, which amino acid is encoded. And then that can have that amino acid being added to many other together chemically can make a lot of different kinds of protein. Very few of them are just one-off. Remember, I mentioned polygyny and pleiotropy. It's everything interacts with everything else, both ways. The trait has multiple single base changes, that make a difference, and the any single base affects many things. So life
Does
is
the
life is not
does the
simple.
term recessive, does the term recessive and dominant gene have any meaning at all? Recessive and dominant.
Yeah, I'm sorry, I couldn't get your question.
He asked, does the term recessive and dominant gene have any relevance at all?
Well, it does, in the sense that you saw that terribly complex network. And these are, when you compare groups who have Alzheimer's with groups who don't. I mean, but does it say more? Well, one day, if we might be able to unravel the whole lot. It seems to me we've moved it with the genome-wide association. We have really moved into a much more complicated world. We should maybe be happy with a gross estimate of heritability.
Jeffrey Milstein also has his hand up.
Yeah.
Yes, thank you very much. And Professor Weir, it's been very interesting and highly complicated. It seems to me that a possible approach for the future, if not already being used, would be artificial intelligence applied to finding the very intricate patterns between the base pairs, the genes, and the traits. Simply because we've multiplied the field of analysis to so many other variables now, because the base pairs, as you point out, are thousands of times more, that is greater in number than what were called the genes in the past. And also the complication that the base pairs are actually affecting protein expression, which themselves affect whatever neurocellular developments in situ occurred. So it seems to me that invariably, if they're not already using artificial intelligence, they will soon need to do so.
Yeah, the Salzburg work, that's where their geneticists are arguing on just the number of what they call genes, or what they call protein coding genes. But you can get single nucleotide differences based on the number of repeats too, which is closer to what we mean by a gene. And you have to go back like to the 3 genetic groups in the Caspi experiment relating stress to depression. But the Salzburg group is the one who have used artificial intelligence to make the count. So yes, there's a lot of people working on that. They're much brighter than I am, all of these people. So Elva, you have a question. But you're muted.
Am I now?
No, I can hear you now.
Okay, good. I mean, there's great hope that manipulating chromosomes, we might be able to eliminate horrible genetic disorders like Huntington's chorea. But on the other hand, some folks worry that it might reach the stage where people manipulate chromosomes to try to produce what they call designer babies. Can you reassure us that we're pretty far from that?
Well, I think just trying to understand what happens, we're very far from it.
Oh, good.
Yeah, wouldn't you agree? I mean, if we're really talking about all these base pairs now, maybe a lot of them are just there for the ride, but there's still so intricate connections between all of the different ones. I don't think that there's any chance of a designer baby. The real problem with attempting to change, let's say, the phenylketonuria, they know that it's located on I've forgotten which chromosome. It's one
I
of
wouldn't know anyway.
Yeah, one of the middle ones or something, that it has one base that's different with a person with phenylketonuria. And what's interesting about that is the trouble is, if you were to go in and change it, you'd have to change it in all the cells. The place you have to change it to make it useful would be right at conception, but you don't know that baby's got PKU at that point. The conceptus has PKU. So, you know, there's such huge practical problems with any kind of change like that, even if it could be a simple, straightforward effect that produces an amino acid that fails to metabolize phenylalanine in that case. Actually, that is a particularly interesting disorder because the children, if outgrow it mostly by age 7, there's another aging effect. Why? What happens in the genetic endowment that makes all these age changes?
Thank you.
Okay, I think that's all the questions we have. So thank you very much.
Right. I like this one chat. Maybe some of you have seen it. I don't have to be so concerned about not remembering my genetics course of 50 years ago. I think the real problem is to get rid of it. Thank you.
Thank you.
Thank you. Okay.